whip spiders, water mites, a few other groups. But if I'm left to my own devices, then I always gravitate back towards pseudoscorpions. So I spent the last 40 years working on them. Basically, I did my PhD over in Melbourne on them. I'm based in Western Australia, which is in Perth and in the other side of the country, and yet continuing to keep working on them. So pseudoscorpions are an order of arachnids, and I'll talk about their relationships later on, but they're globally distributed. This is actually a map of taken from a database that I maintain of the world pseudoscorpions, and it shows the type localities only of all the species of pseudoscorpions that have ever been described to date. So you can see they're basically found in all geographic regions. They don't occur, there's not too many of them up in the frozen north or Antarctica, obviously, but anywhere where there's terrestrial habitat, pseudoscorpions seem to have made it there somehow or another. So they're completely cosmopolitan. They do occur all the way down into the intertidal zone, and I'll talk about one of those representatives a little bit later, and to the slopes of Mount Everest. You get them up in the Himalayas, right up into the snow zone, and what they do over winter, nobody's exactly sure, but they're certainly everywhere. They also get out to oceanic islands, and even relatively recently formed oceanic islands. In the 80s, I was on a trip to Krakatoa, which is an island between Sumatra and Java, and we sampled the biota there that was present at the time. And it's exactly 101 years since the cataclysmic eruption of Krakatoa in 1883 had, biologists think, completely wiped out all life on the island and reshaped the caldera. There are 4 main islands left, and a new one in the middle that had popped up in the 30s. That's the active part of the volcano. And there are about a dozen species of pseudoscorpions, about 10 species of pseudoscorpions that had made it out in the last 100 years. And they do that in multiple ways. One is hitching a ride on insects, and the other is probably rafting across on sort of beds of vegetation and just hitting the beach. I also noticed one day when we were offloading food that had come in to restock our supplies halfway through the trip, there was a big Hessian bag of coconuts that had been delivered from Java, which is the closest island. And as we're offloading them, there was a pseudoscorpion walking on one of the coconuts on the husk. So they get around with humans as well. So yes, globally distributed. If you think they're rare, then you probably haven't got a good eye for pseudoscorpions. Years ago, I was visiting the Sierra Nevadas. I went up to the University of California's research station and was talking to the superintendent up there. And he said, "Oh, so what do you work on?" I said, "Oh, pseudoscorpions." And he said, "Oh, no, we don't see too many here. There was one found in the next valley about 10 years ago, and that was pretty interesting." I said, "Oh, okay, well, all right. Well, I'll just pop outside and do a bit of collecting." And 2 hours later, I came back with about 50 specimens of 4 different species. So you just got to get your eye in for them, and they're kind of everywhere. My kids, who are grown up now, they're in their 20s, they became very, very good little pseudoscorpion collectors, especially when they were in primary school and had better eyes than me at looking under rocks. So they've even found new species here in Western Australia when we've gone on family holidays, which involve dad searching for interesting localities for pseudoscorpions. Pseudoscorpions do come in a range of different colours, all based on brown. So there's dark brown and light brown and brown and pale brown, mottled brown, stripy brown, but mostly just brown. So these are various different families. You can see they're all, they look a bit different to each other. You can easily tell the difference between some of the families. This is one of the most spectacularly modified families. This is the Fielidae. Doesn't occur in North America. There's a single species that we've described from Brazil. They're in Australia, Asia, Africa, Madagascar, and there's some fossil species from Baltic amber and also Cretaceous ambers, which I'll talk about a little bit later as well. These are highly modified. We don't know what they do with these little these little pedipalps at the front here, which are quite small in comparison to other species of the of Pseudoscorpions. So I've been really lucky to be able to travel around the world and collect Pseudoscorpions in a number of different places. So I've done fieldwork in East Asia, Southeast Asia, Africa, North America, a little bit in Europe. That dot there is not a collecting record. That is actually 0 degrees north and 0 degrees east. So Pseudoscorpions don't occur out in the Atlantic Ocean. Well, they do on islands, but not on that place. So that's spurious. So I don't think that I've been to a non-existent island in the off the coast of Africa. So yeah, I've been able to pursue this, my research in various places of the world. As you can tell, the closer you get to my house, which is down here in the corner of Western Australia, the greater number of records that collecting records that I've made. That been to some pretty interesting and crazy places over the years. Pseudoscorpions have been known since antiquity. Aristotle was aware of them. He talked about other organisms that he says they were like scorpions. They have no tails and are very small. And he was talking about a species that occurs or used to occur regularly inside libraries and in collections of books. And this same species was recorded in the 1660s by Robert Hooke, who was the first Englishman to perfect the use of a microscope that he'd learnt from the Dutch who'd perfected the technique. And he studied all sorts of small things, including this little pseudoscorpion, which is likely to be exactly the same species that Aristotle had. And this species is called Chiridium museorum. Its museorum refers to the fact that it occurs in museums or libraries, and they regularly feed on Psychoptera, on sosids in those libraries. They're much less common nowadays because the glues that are used in books tend to be synthetic. They tend to not be the same sort of biological basis that were used in the past, that sosids would feed on, and papers that sosids would feed on. And of course, there's fumigation as well inside libraries. No librarian would want to tolerate an outbreak of something like a sosid and then its predator, the pseudoscorpion, chasing after it. But he said that they had claws that seemed to grow out of the head like the horns of other animals, like crabs or lobsters' claws, and a pair of claws or pincers, which this little animal did open and shut with pleasure, because the claws are like a scorpion's claws. You can see his good little drawing here. So they've been known for a very, very long time. Linnaeus was the first person to formally describe one, like plants and animals. He got in pretty early, and of the 4,000 or so species that he described, he recorded Cancroides from Europe and a species he called Scorpioides from Suriname. Unfortunately, he thought that they were mites, and he put them in the genus Icarus. So he was a little confused by all sorts of things. If anybody looks at his classification nowadays, you'll see he's made all sorts of weird and wonderful mistakes. But this particular species here, which is now known as Cheilopha cancroides, is very, very common in houses and other situations all throughout Europe, North America, parts of Africa, not very common in Africa, one place in Australia, and it's been distributed around the world by humans. We don't know where it's originally from, but Linnaeus was certainly aware of it, and they've been recorded as feeding on bedbugs inside houses. In fact, there's lots of records, lots of specimens in museums, particularly up until about the 1950s, and particularly in North America. Some of the collections that I've studied, lots of specimens of Cheilopha cancroides collected from inside houses. Sometimes it's mentioned so that they're in association with bed bugs, and they seem to be diminishing in houses in places like North America and Europe with the widespread use of pyrethroids, insecticides to kill bugs around the house, and the pseudoscorpions kind of got knocked down as part of that. And now that bed bugs are coming out, a couple of people have even bed bugs are becoming resistant to pesticides and becoming more prevalent. People have talked about reintroducing pseudoscorpions back into houses. I'm not sure they're going to be enough of them to actually keep bed bugs under control, but it sounds pretty good. But if anybody wants to employ me as a consultant on this, I'll gladly go and tell them what I know. So this is Linnaeus's description. In Latin, he called these pedipalps antennae. He said they were cheliform, which just means they form chelate appendages, which just means that the fingers go up and down the end here, and they've got a flat oval abdomen. So well done, Mr. Linnaeus, you nailed it. You covered all pseudoscorpions because that's exactly what they all have. So not a very good description in modern terms, but certainly they've been known for a long time. So I guess the grandfather of modern pseudoscorpion research was Joseph Chamberlain. He did his PhD at Stanford University under the tutelage of Gordon Ferris, who worked on insects, on homopterans mostly. In fact, Chamberlain did his master's on scale insects, on lac insects, and the classification that he devised as part of that study is still basically the same one that's in use today. He switched to pseudoscorpions to do his PhD, and that large monograph that came out of that was published in 1931, and it's like our Bible. We go back to it when we need to find something out about many different aspects of pseudoscorpions. He was based in Oregon for the most part of his professional life. Up at Pacific Grove, there was an agricultural research station that he was based at, and in fact he's buried in the local, his ashes are cremated in the local hospital. So he died in 1962. So I was nearly 4 years old when he died, so just kind of missed out on meeting him, but he sounds like a remarkable individual. His collection is now at the California Academy of Sciences, where Darryl was based for many years, and it's a collection that I visited several times because it includes so many important things, specimens. So this is the classification that he derived with kind of a phylogenetic tree that he devised. He didn't call it a phylogenetic tree, but basically here's the order, which he called Colonophidae at the time. Here's the 3 suborders he recognized. Here's all the here are all the different families. And if you kind of squint, this tree is pretty close to what we recognize now using all sorts of different techniques, which we'll go into now. So I've published with colleagues and myself individually over the last 30 years, different phylogenetic trees based initially on morphology, then on sequence data based on 3 genes, and then more recently, 2-3 years ago, using transcriptomes, which enable us to get hundreds of gene regions out of an individual specimen. And this is the classification that we published at the time. It still incorporates some of the names that Chamberlain himself coined and used, such as heterospheronida, which means different feet, homospheronida, which means similar feet, based on the number of tarsal segments on the various legs. But there have been some new ones as well. So I recognized a suborder in 1992 that I called the Curator, which means venom hand, because this entire clade that's included in the Curator, so from here outwards, they have venom inside their keel fingers, whereas the other pseudoscorpions don't. And then there's a couple of extra clades here that have got these names on the side. And more recently in this 2019 paper, I separated off this really weird superfamily called the Fieloids into their own superfamily, which I called the Topospheronida, which means odd feet or odd legs because they've got unusual legs, and it fitted with the spheronida endings that Chamberlain had devised because they were so unlike anything else. And in classification systems over the years, Fieloids have flipped around all over the place. People have put them associated with other superfamilies and different suborders. But our work convincingly showed that they were a distinct clade all by themselves between this little family, a superfamily of Chthonioids and the Eocuratans. So that's kind of where it sits at the moment. It might change again as we get better techniques and better sampling, but that's the current classification. So I've just talked about venom in pseudoscorpions, and often you read in textbooks and on Wikipedia pages about how all pseudoscorpions have got venom glands. Well, our study has shown that that's not true. These 2 basal superfamilies, and in including this fossil one, which I'll mention shortly, don't have venom glands in their keel or fingers. It's only evolved once here. And most pseudoscorpions that do have them have venom glands inside both of the fingers. So this is a lateral view of the claws. This is what we call the keeler. This is the fixed finger because it doesn't move. This is the movable finger that goes up and down to grasp their prey. And they have these glands that go out and discharge through a tooth at the end of the tip of the keel or finger. Chamberlain was the first really to notice this because he made wonderful slide preparations and looked at them in detail. So as early as 1924, he published this observation that some pseudoscorpions have venom glands in their keel or fingers. And subsequently, venom glands have been lost from either the fixed finger or the movable finger in individual clades. And we can map that on the phylogeny. But it's pretty rare to lose them. Obviously, there's a little bit of a disadvantage if you do lose the venom glands in one of the fingers, but these are the families that do this are very abundant and very common around the world, and there's lots of species. So obviously they're still able to get by and kill their prey. So it is used for immobilizing and killing their prey. I know of someone who was bitten on the inside of their arm, on the soft part of their arm, by a relatively big pseudoscorpion years ago, and he said it came up in a tiny, tiny little red welt. I've never done it myself. I should get myself sort of attacked by a pseudoscorpion and see what happens. But so if I suddenly die in mysterious circumstances in the field in Western Australia, you people will be the first to know that Mark tried to envenomate himself with a pseudoscorpion that didn't end well. Make a funny stories one day. And a few years ago, I was associated with another analysis using, once again, transcriptomes to investigate pseudoscorpion venoms. And they have a unique set of enzymes and protease inhibitors in their venoms that have evolved separately from all other arthropods. And I'll talk more about that a little bit later on. But this is the species that we studied. This is actually a species that's endemic to a small mountain range here in Western Australia, a species I described about 30, 40 years ago. And we did a really nice study on those. But so most pseudoscorpions have got venom. If they were a foot long, we'd be in deep trouble, but they're not because they're tiny. So I talked about the fossil group in that phylogeny I showed earlier, and this is the fossil in question here. This is a thing called Dracocheila that is in its own family, and now we've placed it into its own suborder, which I called the Paleospheronida. That trope of using Spheronida as the suffix paleo, simply meaning old, obviously. And it comes from the Gilboa shales in New York. These are beautifully preserved shale sediments from the Devonian. They're about 380 million years old. And essentially this pseudoscorpion, the bits and pieces that have been studied, essentially look like modern pseudoscorpions. In fact, the trichobothrial pattern, the pattern of the hairs on the keel or fingers, is no different to species of this family, Pseudopteranodontidae, which are still present in the world today. There are other differences, but there are many, many similarities. So pseudoscorpions have been around since the Devonian, and that, to me, that makes them one of the really interesting arthropod groups to work on because they're super old. As part of this phylogenetic study we did 3 years ago, we were able to date a lot of these evolutionary events back to individual families that probably evolved in the Mesozoic or earlier. And we did this by doing a time-related analysis on the data that we had and calibrating some of the divergence points on known fossils. And we have them from basically 3 or 4 different parts of the world and different time zones. So we have Dominican and Mexican ambers here, which are about 20 million years old. We have Baltic and Rovno amber from Europe, that's about 44, 45 million years old. We have a record from Canadian amber, but we're not certain of the identification of the species. That's 70 to 85 million years old. And some of the more interesting fossils are from Burmese amber, mostly from Myanmar. And it's about 99 or 100 million years old. There's a few fossils turning up in other parts of Europe, France and Germany that are the same age. And what you'll see here, these circles representing the presence of individual species that are being recorded from each of these families. So we've got coverage from about half the families now. And you'll see, apart from Dracocheila from the Devonian, every single fossil can be placed into its own, into a modern family. So we haven't seen anything that's an extinct family yet. In fact, some of the fossils I've examined from the Burmese ambers, if they weren't in amber and weren't, people tell me that they're not 100 million years old, I'd look at them and go, oh yeah, I'm sure I could go to a local forest in Southeast Asia and collect that. They're so similar to each other. We can put some of them in the same genus as modern pseudoscorpions. So it's pretty clear that there's really marked in for a lot of groups, there's morphological stasis. They basically haven't changed their morphology for 100 million years. Others have, but some haven't. So that makes it pretty interesting as well. So they're super old organisms. So these are three different representatives that we've been studying from Burmese amber. This was a new genus that we described a couple of years ago, which we named after Peter Weigelt, a very famous biologist who just passed away recently, in honor of his contributions to arachnid phylogeny and embryology and behavior. This is a member of the Idioronchidae, which is a family that's still around today. This was also a new genus. And this is the oldest known representative of the Fielidae, that family I was talking about before with the funky little pedipalps. And they were around in Burmese amber as well. So super old animals. Now, I talked before about how some of the pseudoscorpions that we recorded from Krakatoa when I was out there in 1984, probably got out there by hitching a ride on insects. So we actually do have a lot of records of pseudoscorpions that undertake this behavior called phoracy. This is where they attach themselves to other organisms, often flying insects. And it's a method of moving from one area to another. And there's been a few studies to try and work out why they do it. It's possibly actually used to move from one habitat to another. So they're actually doing it quite deliberately. But some of you as entomologists might occasionally see a pseudoscorpion attached to a flying insect. Sometimes they get in weird places, like the top of the body on this, I presume that's an assassin bug, attaching to the legs of things like flies. This looks like a crane fly. There's a species in North America that only attaches to the setae up near the head in a particular species of moth. You get them attached to flying insects. When in houses, sorry, I should have finished my sentence first. When our eldest daughter was born, I had her on my lap. She was only a couple of weeks old, and a fly landed on my leg, and I had a pseudoscorpion hanging onto the leg of the fly. And the story that I said at my daughter's 21st birthday to all the people who came to her birthday party was that I just about threw the baby to my wife. Here, catch Francis while I go and try and find this pseudoscorpion. So the pseudoscorpion got collected. I didn't injure my daughter, and it's now in the collection of the Western Australian Museum. It's an introduced species that's been transported around the world. It mostly lives on flies that are things like stable flies, Stomoxys, and on musca. So it's been carted around by humans, probably originally from Africa, although we're not sure. So this is really interesting. These records are quite not uncommon to see them attached to flying insects, but pretty cool when you see it. And behaviorally, pseudoscorpions do other unusual things. So they have mating that's usually via some sort of mating dance in the Cheilopharoidea, which is a family that's composed of 4 families. And they have a true mating dance where the males serenade the females. Sometimes he vibrates his pedipalps. I've seen them tap the female's top of her head, and you'll move backwards and forwards, grasp the female. He'll deposit a spermatophore onto the substrate, drag her over it. If she's receptive, she'll take the spermatophore into her gonopore, which contains the sperm. Other families, all the way back to the most primitive family, I don't like using that expression, but all the way back to the chthonioids, they have quite a different technique where they simply deposit a spermatophore on the ground and leave it, and the female finds it and takes it into her gonopore. Then there's various stages leading up to this full mating dance that chelophoroids undertake. And then after insemination, the females have eggs that develop inside the abdomen. They don't lay eggs per se, they extrude a thing called a brood sac. So these are embryos that are being nourished by embryonic fluid that are still connected to the gonopore of the female, and they hatch out and stay with the mother for a few days and then disperse. Now, often all of this is done inside a brood chamber. I can't remember what my next slide is. Done inside a brood chamber, which I'll talk about in a second because there's another slide coming up. There's also ontogenetic changes in pseudoscorpions. So these are drawings taken from a paper of mine I published, a phylogeny paper in the 90s. There are 3 nymphal stages in most pseudoscorpions. The protonymph, oh, there's a typo. Oh no, it's a disaster. That should say deutonymph. Next one is tridonymph, and then the adults. And they add these trichobothria, which are these sensory hairs on the chelar fingers. This is this lateral view of the chelar. And you have a 3-1 situation, 3 trichobothria on the movable finger, 1 on the fixed, sorry, 3 on the fixed finger, 1 on the movable finger, 6-2, 7-3, 8-4. It's very consistent. There's very few deviations from this pattern. And even this Devonian pseudoscorpion that we know of, there's a tritonymph and a protonymph, and they have these trichobothria in exactly the same positions as some modern pseudoscorpions. The positions of them does vary slightly between different genera, different species, and so they are used taxonomically quite a lot. Yes, 4 post-embryonics in stars. There are a couple of genera, a couple of taxa that have undergone neoteny, where the adults actually have a pattern of 7-3, but that's pretty rare. Mostly it's this consistent pattern. So they're one of the few taxonomic groups, including things like mites, where you can actually very confidently state which nymphal instar you have based on the number of trichobothria. This is the silken molting chamber that I was talking about. These are pseudoscorpions on the underside of a rock. There's an individual chamber. It's had its lid sort of, or the silk torn apart as I've lifted the rock up. So this is a species here in Western Australia. And they're extruded from the chelicerae, which are on the front of the body. And there's a thing called a galea here, and that discharges, excuse me, into a silk gland in the head region here. Excuse me. And they extrude that silk and turn that into a little silken chamber where they spend their life. Sometimes they secrete themselves in there. Intertidal pseudoscorpions will actually make one of those or retreat back into one of those every time the tide comes in to stay secure. The tide goes out and they start moving around looking for prey. Tide comes back in, they get into their silken chamber with enough oxygen to keep them going for a little while. In terms of diversity, as I said, they're found all over the world. Last count, there's nearly 3,800 species in about 470 genera, 24 families that we currently recognise. They're found all over the world. Now, I didn't have a chance or time to update this graph, but this is a graph taken from a paper of mine in 2007, shows the number of recognised species compared to the log area, square kilometres of the country from which they're been recorded. So it's an indication of diversity of habitat as much as anything else. Russia is very low, even though it's got a very big area, because so much of its landscape is unsuitable for huge diversities in things like pseudoscorpions. The USA is at the top of the list right now, largely because there have been so many people working on them and a huge diversity of landscapes from the north to the south, east to the west. A lot of cave systems, turning up lots of new species. I'm single-handedly trying to get the Australian number of species up over that of North America, but I'm going to have to keep working a little bit harder to get there. But you can see that you know, a lot of research work that's been gone in Spain, Mexico, Brazil, etc. But that's just an indication of diversity. Now, in terms of relationships, pseudoscorpions have often been thought to, for a long time, were thought to be most closely related to solifuge or the sun spiders. But it turns out that those morphological data that we use to make that relationship might not be all that reliable. We've tried, or people have tried over the years, to use standard genetic molecular techniques to look at relationships among arachnids. And each of the orders of arachnids are potentially go back as far as the Devonian, based on things like pseudoscorpions. We have mites also from those Devonian shales. So they're super old groups. And so the molecular signature becomes very confused and very uncertain. This is a paper that we published just recently at the end of last year, using a different set of data than just regular sequence data and comparing them against each other. I'm not going to go into the details because largely I probably can't explain them as well as some of my other co-authors, but what they tracked here was this thing called whole genome duplication. And so there's a whole set of gene regions that have been duplicated across the genome in some areas of some arachnids. And looking at every single order, it turns out that these duplications have only occurred in this suite of species here. So this includes spiders, the Arachnidae, whip scorpions and whip spiders, Europygi and Amblypygi, the schizomids or the micro whip scorpions, which aren't here in this photograph. But they've also been shown to be found in scorpions. And that's one of the reasons that recently scorpions have been added to this clade that includes the tetrapulmonata, which are the spiders and their relatives. New work that's been done in conjunction with one of the labs in the US has found that pseudoscorpions share exactly the same gene duplication. And it looks as though they're sister to scorpions, which explains why they've all got chela that look like this, and just pseudoscorpions are missing the tail and the pectines that scorpions have. And there's a range of other things that are only found in this clade as well, although not uniquely. So book lungs are only found in representatives of this clade, but not in pseudoscorpions. The mating dance, the promenade deux, is found in some pseudoscorpions, but all scorpions, all whip scorpions, all whip spiders. A stalk spermatophore is found in all of these groups, except the spiders. And a brood sac from the genitalic glands, the female glands, is found in a bunch of them, but not all of them. So all of this evidence leads us to think that pseudoscorpions and scorpions are closely related to each other, which is what was first thought back in the 1800s, but by the early 1900s wasn't the case. So we've come full circle, which is kind of nice. And for people, I know some of my colleagues don't like calling them pseudoscorpions because it says they're false scorpions and they're not actually scorpions. Well, we can go back to using that name quite happily now if pseudoscorpions and scorpions are thought to be sister taxa, which seems to be the case, at least currently. So in terms of collecting, if you're interested in finding them, occur under rocks, under bark. They also occur in mammals and birds' nests pretty frequently. As I said, you can get them from insects, sifting leaf litter, or using Belize or Togolin funnels or Winkler sacs here is a great way. I just about haven't been anywhere in the world where I haven't been able to find at least some pseudoscorpions. In highly urbanized areas, you can't, of course, but any natural ecosystem, they're just about everywhere. So if you're interested in taking a look at them, I sift leaf litter in the field into a white tray, sit there on a log or a rock and just wait for them to start moving, then pick them up with a paintbrush or a pair of forceps. but they're pretty ubiquitous. In some areas you can get, you know, a dozen species, probably the best areas I've ever collected diversity have been in parts of Southeast Asia. You can get 15 species in a single site, you know, collecting pretty hard for 2 or 3 hours, sifting leaf litter and peeling bark. And often that's a very, very different fauna. Different groups will be in leaf litter compared to under bark of trees. Canopy fogging is used in some places as well. You get canopy pseudoscorpions. I've never done this myself. I don't think the licensing agencies here in Australia would allow me to use a technique like that, but I've seen people use them. And just my last couple of slides are about classification. So pseudoscorpions are grouped into, oh, interesting array, into what's that, 1, 2, 3, 4, 5, 6, 8 different superfamilies. There's 24 living families that I mentioned before. The 25th is that Devonian one that's extinct now. And in California, there are, oh, I forgot to count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. There are 16 families, sorry, there are 15 families currently represented, known from California. So I thought I'd, you know, localize it and help you guys out. The only one that I've included on this list that actually technically hasn't been found in California yet are the Stenophoridae. These are very flat pseudoscorpions that are found in various parts of the world. They occur in northern Mexico, pretty close to the Californian border. So I've included them on this list because I think they're going to turn up at some stage. So there are species that are known and also others that are known from Texas and further to the east. Quite common in Florida. They'll probably turn up one day. And they've been found most frequently under mesquite bark. So if you feel like peeling a bit of mesquite bark and you find a stenophorid, give me a buzz. I'd be interested to hear about it. So yeah, it's a diverse fauna. You get all sorts of things, families that are quite common, conetids, which are a very diverse family all over the world. Some that are very, I've already I've counted them up. Here we go, 16 families in California, 47 genera, 91 species, but there's lots and lots and lots of new species. And these are the number of genera on the right-hand side. So some are quite rare. This family with the big mouthful name, Pseudotrenchothroniidae, there's only a few records from Northern California. Others like Neobasiidae are very, very common. Cothoniids are very common in leaf litter. Gyripids are only found down near the seashore. So there's a lot of diversity and for people who are interested. So just to finish up, I wanted to talk about a project that I have been doing for the last few years, thankfully with Daryl's help, which has been absolutely fantastic. We've been looking at this family Pseudogyripidae. Now this is the known distribution of Pseudogyripids around the world. The red circle here are a few named fossil species from Baltic amber, so that places them about 45 million years old. The only other places in the world that they've been found is one gorge in Tasmania, just outside the little town of Launceston, and Pseudogyripids, it's its own genus down there. They're very common. And then you get them in the Rockies and you get them up in northeastern USA and southeastern Canada. So I've been working on started on the Californian species. We've got a lot of sequence data now, and we've got a lot of cave material that Daryl's been fantastic at collecting and sending over for us to study. And I'm super confused at the moment. So I think there's lots of species. Certainly seems to be quite a few cave species. This is one of the cave ones. It's got slightly longer, slightly thinner pedipalps than the ones on the surface. They're not full troglobites. They've still got eyes, but they're paler. They look different to the surface species. And then of the surface species that have been classified as Pseudogaripus bicornis in the past, which was meant to range all over the Western USA, we're getting some really big genetic differences between them. And so I'm going back trying to see if I can find morphological differences, and I occasionally can. So if you happen to find a Pseudogyripids genus Pseudogaripus in your travels, if this looks familiar to you, I'd be really interested to take a look at them. I'd be interested in doing more sequencing, because I'm a bit confused right now. So I've got some more specimens coming from Harvard University, and we'll do another round of studies and see if we can get to the bottom of it. But they're really cool. If you see one of those, you're pretty lucky because they're pretty funky animals. And up close, all pseudogryphids have got 3 lobes on the front of the carapace and eyes that are set back here on the carapace. So under a microscope, they're pretty easy to, even with a hand lens, you'll tell them apart from anything else. So they're pretty special. And the last thing I wanted to talk about was the family Gyripidae. In California, there's only representatives of one genus, a new genus we described a couple of years ago, taking some species from California, Mexico, and Japan and Korea and putting them into their own genus. They've sort of got a triangular carapace, eyes removed from the head like the pseudogryphids, but they are found only down on the seashore. So you see them you find them under sea wrack and driftwood, sometimes rocks, and they're really nice as well. So if you see those, you're pretty lucky as well. Thank you for your time. Thank you very much for inviting me. And I'm very, very happy to answer any questions. I'll stop sharing my screen, so thank you.
Well, thank you very much, Mark. I enjoyed that. I hope everyone else did. And I will see if I can contribute something to the world of pseudoscorpions myself, if I can ever get around to looking at some of my old stagnant collections. Maybe that's one of the reasons I was trying to get in touch with Daryl. I was hoping to mobilize some of my material. And we greatly appreciate your talk. Were there any other questions? Anybody?
What do you think the percentage of cave species compared to non-cave species are?
That's a very good question. It's a very good question. In some groups, it's highly skewed. So you get whole groups of species that nowadays only occur in caves or other subterranean environments. But in general, not all genera go into caves. That's not part of their shtick. But maybe, if I had to say a figure, maybe sometimes 10%. But there's huge radiations of cave-dwelling species in places like Mexico, California, and I see it's Rolf who's asked the question. You are famous in pseudoscorpion circles. You've collected some amazing pseudoscorpions and had a couple of species named after you, I think, like Daryl has. So it's my absolute
a type genus.
Oh wow, yeah, that's nice. So yeah, you do get, there are quite a few groups that end up in cave systems. There's a genus called Tyrannochthonius, which is found around the world in leaf litter and caves, and we've got a huge radiation of undescribed species here in Australia from caves, and many from leaf litter. And in the southeastern USA, Alabama up to, I can't remember the other places, Tennessee, Texas, there's dozens and dozens of species that have been described, each species found only in its own little cave, but very few epigean species of that genus in southeastern Australia. So it's probably some older radiation that's been extirpated on the surface, but they've survived in caves for some reason. So it's highly variable, Rolf.
So one more question. You were talking about both scorpions and pseudoscorpions being Devonian in origin, but do you have, is there any evidence of which is older?
Yeah, there are older scorpion fossils. I think I'm a bit hazy on this. I think they go back to the Silurian. Okay. So scorpions are older. And I'm not sure about, I'm a bit hazy about scorpions. Some of those really early scorpions might have been aquatic. So the other thing I didn't mention in all of that is that we've just published or had accepted a paper which also uses these fancy genomic techniques and places Limulus, the king crab, Xiphosurans, firmly within the arachnids. They're not sister to the rest of the arachnids. So this like, yeah, which is like, and it's been coming up for years. People have just been going time and time. It's kind of super weird and it's kind of super counterintuitive, but it's also, it probably means that terrestrialization has occurred in arachnids on more than one occasion, which is super interesting.
Mark, I was wondering, a few years ago, we collected some, I thought that they were some garipids that we sent you from Arizona.
Yeah, you did, Gillian, and I
I don't want to put you on the spot, but
no, you have. So well done. I saw your name and went, "Oh my God, those samples that Gillian sent." So I should now say, "Oh, our connection's really bad. You're fading out," and then run. But look, they're a different family that's got a similar name. They're Garapinidae, which is
okay.
Genus is Cerianus. I will
yeah, there was another one though. There was a weird little thing that was real pale. It wasn't a Cerianus. It was something. Pardon?
Solanus?
No, it was a whole nother thing. I have them shown in my book actually, but
Oh, do you? Oh.
Yeah,
I've
yeah.
got a copy
So
of the book, but I haven't.
If you've got a copy of the book, you can look in there and see those pale
Yeah,
things.
I will.
They were found like on ironwood trees, believe it or not. They were low elevation, and the closest relatives were like in Texas and then Mexico. And so we didn't know which group it was. It was something where they were like hundreds of miles away, the closest relatives, whether you went to the Texas one or the Mexican one. So okay. I didn't want to put you on the spot, but
no, it's a good,
you're a captive audience right now.
Yeah. I can't remember what that might be, but my memory's getting worse as I get older. So I'll look into it. No, I'll promise I'll get back to you.
Okay.
Yeah,
and
they're cool.
So are you still interested in receiving that sort of stuff, or we kind of lost connection back a few years ago, and I haven't really been collecting them. But
yeah, I got sidelined onto the redevelopment of our new museum, which opened a year ago, and I was full-on display person for a couple of years, and so a lot of things withered on the vine. Look, let's talk offline. I'm always keen to look at pseudoscorpions, but what I can't promise to do is work them up and turn them into a scientific paper, because I'm stretched in every single direction. But I'm very keen on samples that are freshly collected, especially into 100% or 95% ethanol, so we can sequence them, because it's more easy for me to drop them into bigger phylogenies. So that would be really interesting. But for any of you, look, I'm always willing to take a look at a few photographs that you might take, and I can give you pointers on at least the family that it's likely to be, and sometimes the genus. I'm always happy to do that. I actually really enjoy it. But if you send me samples and I'm going to say, oh, that's really interesting, and then like poor Gillian, I probably haven't looked at those specimens again in the last
Don't worry about it. I know how things happen. You know, people get kind of busy. Yeah, you only have a couple hundred trapdoor spiders to describe. I mean, chop, you know.
Yeah, but I want to get back onto pseudoscorpions. I want to describe. I got so many new species, you know, and I'm pretty keen to. The trouble is, I'm a perfectionist as well, so I turn my papers into. Well, they're not works of art. I try to turn them into works of art. It's more like finger mud drawing, but I spend a lot of time.
So Mark, in the past, I've always sent my pseudoscorpions to you much more. And I'm sure you're collaborating with him quite a bit, or?
No, Bill passed away about 4 years ago.
Right.
He was super old. The last 10 years of his life, he was in a kind of a nursing home after his wife died.
Okay.
I knew Bill extremely well. I only ever met him once in person. I actually stayed at his house in 1989. He got me. How interested are you people in anecdotes? I really wanted to meet him, so I went to Rochester, and I caught a flight from. I had no money, so I caught a bus from New York to up to Rochester, which on the map looked about this far, right? And it turned out that it was about 5 hours, and I was really badly jet lagged. I booked into this hotel, and I was offered by the concierge when I checked in, I was offered to have a friend stay over the night if I wanted. And I'm so tired and so jet lagged. What do you mean? I don't know anybody in town. He said, no, if you want a friend to stay over, it will be an extra charge. And I finally worked out what was going on. I went, no, don't do that. So I checked into this room, and there were tiny little cockroaches all over the bed, and the sheets were stained, and I was so tired, I just crashed. I went to the university the next day, and I tracked down where Bill's office was. I said, Bill, I'm here. And so we talked. He said, where are you staying? And I can't remember the name of the hotel, Empire, something hotel. And he literally went white, and he said, oh my God, that's the worst hotel in all of Rochester. I said, well, it was really, really cheap. He said, no, I'm checking you out of there. And he drove over with me and dragged me out of this hotel, berated the concierge, and then took me to his house, and I stayed there with him and his wife for a week. It was just lovely. It was just such a lovely man, and we collaborated on a couple of papers. But his collection now is in Florida at Gainesville, but there's nobody working on it. Lovely man. He described a lot of species, and he's the guy who named these species after Rolf and Daryl. Sorry, that was an aside. Stop me if you don't want anecdotes.
Do you have any social pseudoscorpions in Australia? I've heard of some in South America, and some even have what appears to be a caste system, which is really interesting.
Yeah, I didn't talk about that. It's a great question, Gillian. Thank you. No, we don't have them, not social ones. The species in South America that Gillian's referring to is a thing called, I think it's called Paratemnoides elongatus. Yeah. And it looks as though it might be the same species from where different people have been studying them. And this behavior was first reported in the 70s in a population of probably the same species in Florida by Vincent, was it Vincent Brack? Is his name, entomologist? Anyway, yeah, so they look after each other. There's a bit of a caste system. Sisters will help rear broods together. I can't remember the details. I have seen exactly the same behavior in a species in Singapore. Well, what I think is the same behavior. Big clusters of them, all grouped around each other, feeding on the same prey item, adults and juveniles doing it. So it might be maybe different species in the genus do the same thing. Maybe it's the same species that's been carted around the world by humans, I don't know. I haven't seen it in Australia, no. But it's this one genus that this Paratemnoides that seems to do it. I've collected Paratemnoides in Northern Australia, but never like that. Only just single representatives, or one or two under a tree.
Okay, well, any other questions?
The magical Zoom silence.
Yes.
Thank you, Mark. I enjoyed your talk a lot. Thank you.
Thank you, Dennis. I appreciate it.